Hi, in this video I'm going to solve another question from IIT JAM 2016, and this question is on cleavage site on polypeptide. So this question asks that the following peptide, they give a peptide sequence and was sequentially cleaved by dithiotritol, cyanogen bromide and trypsin. So in order to answer this question, we need to know that what are the cleavage site of trypsin, cyanogen bromide and dithiotritol. So and then only we can say how many peptide fragments are produced. So let's see a piece of information. So here we have some agents and their site of cleavage, some popular agents that is hot favorite for IITs. One is trypsin. So trypsin always cuts in the C-terminal side of lysine and arginine. That means the basic amino acids. So they would cleave after lysine, after arginine. Whether there is a lysine or arginine, trypsin would cut after it. Chymotrypsin, on the other hand, would cleave at the C-terminal end of aromatic amino acids such as tryptophan, tyrosine, or phenylalanine. So these chymotrypsin would cleave the C-terminal end, that means after tryptophan, after tyrosine, or after phenylalanine. On the other hand side, cyanogen bromide will exclusively cleave after methionine. It would cleave at the C-terminal end of methionine. Now, pepsin would cleave at the amino terminal side of the aromatic amino acid like tyrosine, phenylalanine, and tryptophan. So pepsin and chymotrypsin, their mode of action is almost same, but chymotrypsin cleaves at the C-terminal side of the aromatic amino acid, that means after C-terminal, after these amino acid, and pepsin would cleave before these aromatic amino acids. So just we need, for this particular question, we need to understand the cleavage site of trypsin and cyanogen bromide. So for trypsin, it's lysine and arginine. Whether we see a lysine or arginine, trypsin would cleave after that, and cyanogen bromide would cleave after a methionine. So let's see our peptide sequence. So here is our peptide sequence. So here is phenylalanine, tryptophan, lysine. Lysine is a basic amino acid, so trypsin would cleave it. Then we see tyrosine, methionine. After methionine, we would get a cleavage by cyanogen bromide. Then glycine, alanine, cysteine, then proline, then methionine again, and there is a cleavage point for cyanogen bromide. Aspartate, glycine, arginine, again arginine, there is a cleavage point for trypsin, phenylalanine, alanine, glycine, and tryptophan. So there are basic 4 cleavage site, and 4 cleavage would produce 5 fragments. Now the thing to be noted that don't get confused with the function of dithiotritol. Now, what is the mode of action of dithiotritol and what is the cleavage site of dithiotritol? So dithiotritol basically cleave disulfide linkage. So if there is a basic form of disulfide linkage, dithiotritol will cleave this bond, but it won't cause fragmentation of the peptide. So here in our structure we have a disulfide bond between these two cysteine residues, and it would be cleaved by this dithiotritol, but it won't produce more peptide fragment. So how many peptide fragment we are getting? We are getting basically 4 peptide fragment. Let's label them. So this is our first peptide fragment, this would be our second peptide fragment, and this would be our third peptide fragment, our fourth peptide fragment, and this would be our fifth peptide fragment. So the correct answer would be 5. So this question came under the numerical ability section, NAT section, and if you do this question correct, you would get a 2 marks. And this question is very easy and very important for other competitive exams. A lot of competitive exam ask this type of question. So you just need to understand these cleavage sites. So basically it's good to remember all these cleavage sites because question generally comes from these type of enzymes. They can also ask about chymotrypsin or pepsin. They can digest the fragment with chymotrypsin or pepsin and then say how many fragments would be produced. Okay, and this concludes the video. Please like and subscribe for more videos. Hope you enjoyed it. Thank you.